Welcome to gene regulation. Today, gene regulation will be described and two examples of it will be visualized in prokaryotic organisms. Gene regulation is the cell's way of determining which genes need to be turned on or turned off, depending on what the function of the cell is in the organism. This regulation is crucial to the survival of the organism, since if all of the cell's genes were constantly turned on, the organism would run into many problems in its systems. However, there are genes that are always turned on in the cell. These genes are called constitutive genes and are also known as housekeeping genes. Examples of these genes are if they code for the cell structure or basic functions. For the more specific genes, they usually require activation or suppression in respect to the cell's needs and/or functions. This process was discovered around 1960 by two men named Jacob and Nahad. They discovered how gene transcription was regulated in prokaryotic organisms. They labeled this mechanism the operon model. An operon is a fairly simple DNA sequence containing four primary areas. First, there is the promoter region, where RNA polymerase attaches to and begins transcription of the genes ahead. The structural genes ahead, which code for the necessary proteins for the desired function. The operator, where a protein called the repressor binds to and does not allow RNA polymerase to transcribe. And finally, we have the regulatory genes, which code for the repressor that was previously mentioned. If the repressor does not bind to the operator, transcription occurs as usual. When the repressor does bind to the operator, no mRNA is transcribed from those genes. Activators can also bind to the sequence, allowing RNA polymerase to transcribe even more mRNA than usual. The two examples that will be visualized are the TRP operon, also known as tryptophan biosynthesis, and the LAC operon, also known as lactose metabolism. The host of these operons will be Escherichia coli, also known as E. coli. The first operon on the menu is the TRP operon, which is supposed to transcribe for the amino acid tryptophan. The TRP operon has 5 structural genes, which would synthesize tryptophan if they transcribed. When tryptophan is absent in the cell, the repressor is inactive in the operon, and RNA polymerase has a nice and clear path to transcribe tryptophan. This process is anabolic in nature, since it is building up amino acids for the cell. When tryptophan is present in high concentrations, it acts as a co-repressor. A co-repressor is something that binds to the repressor, allowing it to bind to the operator and block RNA polymerase's transcription. No transcription occurs in this scenario. Next up, we have the LAC operon, which would transcribe for the degradation of lactose. E. coli tends to favor glucose in their metabolism, so breaking down lactose into its components of glucose and galactose is preferred. The enzyme beta-galactosidase is needed for this breakdown, which needs to be synthesized and coded by the genes. However, when lactose is not present, this enzyme is not needed in the cell, so it should not be produced, or else it would just be a waste of time and energy. The LAC operon contains a 5th area as well, the catabolite activator protein binding site, also known as the CAP site. Proteins can bind here to increase the rate of transcription, acting as activators in the operon. When lactose is not present in the cell, the repressor is naturally bound to the operator, blocking RNA polymerase's transcription. No enzyme is produced in this case. When lactose is present, an isomer of lactose, Allolactose acts as an inducer and binds to the repressor, similar to the co-repressor. However, unlike a co-repressor, the inducer inhibits the repressor from attaching to the operator, and RNA polymerase can transcribe freely. This is a catabolic process, despite the fact that an enzyme is being synthesized. Remember, the goal of the operon is to break down the lactose, which is catabolic. The scenario previously described is when lactose is present and glucose is also present. But when lactose is present and glucose concentrations are low, cyclic adenosine monophosphate, also known as cAMP, is produced. The cAMP attaches to a catabolite activator protein, which allows the cap to bind to the cap binding site. This whole process acts as an activator in the operon and speeds up the mRNA transcription by helping RNA polymerase bind to the promoter region. This produces more mRNA than with higher concentrations of glucose to help produce more beta-galactosidase and accelerate the breakdown of the lactose. Generally, anabolic pathways contain a repressor-based system, where the nutrient is the co-repressor, like how is visualized in the TRP operon. Catabolic pathways usually contain an inducer-based system, where the nutrient induces the synthesis of the enzymes, as seen in the LAC operon.